suggesting that at least there are some major loci that are contributing to this process. And so what I simply did was just genotype loci within these two regions and ask, is there association between those regions and flowering time, and see if this replicated. And sure enough, in one of the populations, we see here's the coastal parent, flowers later than the inland parent. And if you're homozygous for the coastal allele, you flower late. And if you're homozygous for the inland allele, you flower early. And I found this in all 4 of the population crosses. So it looks like that same locus is contributing to flowering time in all of those crosses. It's separating those 2 ecotypes from each other. And overall, we found for that QTL, 5 out of the 5 crosses, we see that. But then for the other QTL, we only found it in 3 out of the 5 crosses, suggesting that maybe it's something that's more polymorphic, could be different loci, could be I didn't have enough power to detect it. So it's still not clear exactly what's going on there. But what's really neat is this same QTL is contributing to flowering time over a 1,200 kilometer region, which is a pretty large region. But what was interesting to me is that we tried to fine map this thing, and we're like, okay, we want to know what this gene is that's causing this effect. And so I went out and I tried to fine map this thing, and all of my markers went to one region, and there was no recombination. And I was like, damn it! Seriously. And so like the main conclusion that we could have from that is that, like, okay, this is due to some kind of chromosomal inversion or some major suppressor recombination. Because this is like about 30 centimorgan region, and it's collapsing down to no recombination. So, want to know, okay, getting ahead of myself. Okay, let me go cross those other populations. Maybe that's just some freakish event, and I crossed the other 5 populations, and no recombination in all of those population crosses. So then I made a whole bunch of other crosses. I crossed coastal ones to coastal ones, inland ones to inland ones, to see if there's actually a reversal of marker order there. And sure enough, we see a reversal of marker order. And by making all these different F2 mapping populations, I was able to map the geography of where the different inversion types are. So one inversion type, which I'll call the P inversion type, was in all the coastal populations going up across North America in these perennial coastal populations. The A type, the annual type, was in these orange annual populations, and also in a selfie population that's closely related to Mimulus guttatus. But the really interesting thing to me was like, okay, I was like, wow, these things are like fixed between habitats, this is really exciting. But there's always been floating in the back of our minds are these inland perennial populations that live in hot springs, along rivers, stuff like this. John's been telling me, he's like, those things are like the coastal ones, they must be related. I'm like, no, I don't even want to hear about it, don't talk to me. So I made some crosses to those, and interestingly, they had the same orientation as that coastal perennial population. So the inland perennials also had the same chromosomal orientation. So we've seen like replicated effects of this inversion. This is that locus that we see in all the crosses. And there was a population that didn't like totally group with either one in structure, and this happened to be one of the inland perennial populations. So I was like, okay, I can explain this. I've got another explanation for this one right here, but some other time. And so Elin in our lab and me, we set out and grew up a whole bunch of populations from all across North America. And she's been doing some work where we genotype these at microsatellite loci, and then she's genotyped them in the inverted region of the genome. And if you look at structure, you have all